Hey everyone, we're talking a little bit about energy today, and we're going to talk a lot about enzymes. Some of what you've heard, we've talked about before, and some of it may be a little bit new. So make sure you have Bill out and that you are following along and making sure you're adding to your notes as you see fit. So when we are talking about energy, we are talking about how does a cell that requires energy get the energy it needs to do all of its daily functions. We looked a little bit about how complicated the cell was, all of the stuff that it is doing every second of every day in all of your trillions of cells. A lot of energy is required in order for all that stuff to be happening. So how does energy get into our cells? You also have to consider the fact that at each trophic level, not all of the energy is available to a cell. So if you look at, for example, down here, we have producers. They are the ones that get energy from the sun, and they have a lot of energy available to them. They basically have unlimited energy in the form of the sun. They're able to make their own food. But when they, whatever energy they have, and they move it, whatever energy they make, and it gets transferred up to the next level, only 10% is going to be transferred up to the next level, to the next trophic level. And the same with the next one. Only 10% of what was left goes up. Here, same, 10%. So say you have 1,000 calories available down here. Since only 10% is going to be transferred to the next energy level, the little grasshoppers, they only get about 100 calories. And then you go transfer it up one more time to this mouse. There's only 10 calories available to Mr. Mouse. And then get transferred up one more time. 10% of 10 is only 1 calorie. So the higher up on the food chain you go, the less available energy there is for you to find. So our cells have to be pretty efficient at dealing with the energy that we do acquire, making sure none of it goes to waste and that we are able to harness all of that energy in a way that our cells need. So now all of those cell functions require energy, and we need to get that energy somehow. So basically, you're looking at a flow, an energy flow, and sometimes you can see this like in a, sometimes it's referred to as the carbon cycle, but basically you have a flow of energy. It starts with the sun. The sun shines on photosynthetic organisms like plants. They take that solar energy, they convert it into ATP and organic molecules. You got to remember that ATP is kind of like money for the cells. It's the energy, it's what it pays to get everything done is that form of ATP. So ATP is the cell's energy, and it also makes some organic molecules. And in the case of plants, we're talking about glucose is what it makes. Then those plants get eaten by, say, herbivores. These giraffes will eat the leaves that have made that ATP and organic molecules. And then the giraffe takes those in and they perform cellular respiration and they make more ATP and different and more organic molecules. And then poor little giraffe here gets eaten by the lion and he makes ATP and organic molecules. And the process continues over and over again so that you could have had one carbon atom that was started in a tree because it took in carbon dioxide. So it used to be in the atmosphere. That carbon atom can travel to this giraffe and then into this lion. And now this is where that carbon atom is. So if you think about it, same thing happens with you. You could have eaten something yesterday that had a carbon atom that came from the ocean at one point. So the atoms that you're eating were cycled. They were probably already gone through somebody else and went through the whole process, and now you're eating it because everything is getting recycled. Okay? So if we look at energy flow in living things, we are looking at the capacity to do work, the capacity of um, the cell to do what it needs to do. It requires energy in order to do that. There's two kinds of energy. We're talking about kinetic energy and potential energy. Kinetic energy is energy due to motion, and potential energy is the stored energy due to an object's relative position. For example, if you have a hill here, then you have a ball at the top and the ball at the bottom, which one has more potential energy? Well, it would be this guy here because he is up top and he has more of a more potential energy because of where he is, because he has the potential to roll down the hill. Thermodynamics is the study of energy. And anytime we're talking about studying energy, we're talking about thermodynamics. And there's lots of ways that people go into this field. People go into this field when they want to look at fuel sources for like houses and cars. People go into thermodynamics when they're looking at athlete performance. They want to help athletes be able to utilize energy most efficiently, so that they can take in the least amount they need, but still have enough to have optimal performance for their sport. So that there's lots of different ways people use thermodynamics in their careers. A common unit of energy that we'll be using is kilocalorie, and all that is the calorie that we normally use on a daily basis. If you look at the bag of chips you have, and if it says it's 100 calories, what it means, what it's talking about is that actually it is a 100,000 calories. It's only 100 kilocalories because the calories we use, instead of calling it kilocalories, people just started in everyday language calling it a calorie and it stuck. So when you're looking at your bag of chips, those are actually kilocalories. And they define a calorie as the amount of heat required to raise the temperature of 1 gram of water by 1 degrees Celsius. Okay, so enzymes. We've talked a lot about them so far the last couple of days, but basically this is a refresher of what we know already. Enzymes are proteins. They are biological catalysts that speed up biochemical reactions by lowering the activation energy required. They have a tertiary shape that gives it its function. If you remember, the primary shape was the order of amino acids, and that was primary. And the secondary shape was if it bent into beta sheets or helixes. And then the tertiary shape was its three-dimensional shape, and how it actually folded up. They have a region called an active site. So in this enzyme here, that would be the active site there. It is the part on the enzyme where the substrate is going to bind. Active sites are very specific to the substrates that they're going to act on. Most of the time, nothing else can bind there, or nothing else is meant to bind there anyway. They're specific to that particular substrate. And enzymes are reusable. That's something that we really haven't talked about, but it's a good point to make, in that the enzymes don't get wasted. They're like, think of them like a tool. They don't get used during the chemical reaction and then we need more enzymes. They're more like a tool. They can be reused over and over and over again. As soon as it's empty, like another substrate can bind and it can do it again. And then it can find another substrate and bind and do it again. Kind of like our hands when we were doing the toothpick A's lab. We didn't have to use new hands every time. We use the same hand to kind of grab new toothpicks over and over and over again because enzymes are reusable. Enzymes can be turned on and they can be turned off. And we'll talk about that in a second, about ways that you can, that the cell uses to kind of turn on enzymes and turn them off depending on what the needs of that particular cell are. I want to point out too here that the enzyme, when the substrate binds, this is an example of a substrate that is going to be broken down by the enzyme. Enzymes, remember, can put things together and take things apart. So in this example, they're taking it apart, and basically because of the shape of the enzyme, when the substrate binds, you can have this tugging on the substrate to put some stress on this bond here that's holding those two things together. And once that is broken, then you have two products and they can go about their business and go on their way. And then another substrate can bind to the enzyme. So we also talked about how names are often indicate the substrate involved, like pectinase breaks down pectin, lactase breaks down lactose, maltase breaks down maltase. Um, these are examples of it, but they're not always the case. Like amylase, you find this in saliva, and it breaks down starch. And so this chart gives you the enzyme, gives you its substrate or what it acts on, and then it also tells us what the products are. So lots of times you can have this different enzymes like maltase and cellulase. They both work on different substrates, and our product is the same here in this case. It's glucose in both cases. They're breaking down um, polysaccharides or disaccharides into their individual glucose molecules. So catalase is one that is pretty important to the cell. Catalase breaks down hydrogen peroxide, which is H2O2. This is a balanced chemical reaction for H2O2 with an enzyme. H2O2 can be broken down into H2O and oxygen. Now, in a cell, hydrogen peroxide is pretty toxic, especially with it at high levels or very concentrated. You probably have come into contact with hydrogen peroxide, like maybe if you had a cut or something and mom put hydrogen peroxide on you and it bubbled up and because it cleans wounds. But that's very dilute hydrogen peroxide. Most of the time, that peroxide that mom uses on your little cuts and scrapes, that's only 3% hydrogen peroxide. So it's very, very dilute. If you have more diluted, or I'm sorry, more concentrated hydrogen peroxide, it actually kills cells. This is a picture of someone whose fingers have touched hydrogen peroxide, and your fingers turn white. And sometimes it's lots of little dots. And basically, that's because the cells have died. And eventually, what happens is the skin will get dry and then it'll start peeling off. Sometimes it hurts, sometimes not. It depends on how much hydrogen peroxide that you get on your fingers. I've done this to myself several times using it to clean stuff or whatever. But so it's not great in the cells. So if you can imagine inside of our cells, hydrogen peroxide is actually a byproduct of a lot of the chemical reactions. So since we have a lot of these chemical reactions in the body, we have to find a way to deal with it. The cells have to find a way to get rid of it. So they use catalase. Catalase is an enzyme that will break down hydrogen peroxide and make this reaction go much, much faster. To the point of, I looked this up and did some reading. Catalase can break down 40 million peroxide molecules every second. I mean, just think about that. That is happening every one of your cells. Think about how fast that is. How many hydrogen peroxide molecules that is. I mean, it's just crazy to think about and boggles the mind. This is a picture of catalase and picture of hydrogen peroxide. So basically, that big honkin' protein is designed to bind with the hydrogen peroxide and break it down into hydrogen peroxide, or I'm sorry, water and oxygen. All right, activation energy. When you have a chemical reaction, you're talking about this amount of input that you have to put in first. So this is the activation energy. It's the amount of energy that you need in order to get past this point of no return so that the chemical reaction can proceed. If you don't put in this initial amount of energy, the chemical reaction won't happen. Okay? So it basically is going to destabilize the existing chemical bonds. And that's what the activation energy is doing. It's destabilizing those bonds to allow the chemical reaction to take place. Most reactions require energy to get started, and catalysis is the process of lowering necessary activation energy. So anytime you're talking about catalysis, you're talking about using an enzyme to lower the activation energy for a chemical reaction to proceed. So here's what we were talking about in a prettier picture. So you have the reactant and you have products. You have this hump here. This is the amount of activation energy required to put into this chemical reaction without an enzyme. If you add an enzyme in it, and then now it is called a catalyzed reaction because you have an enzyme in there, it lowers the activation energy, and now much less is required in order for the reaction to proceed. And so the reaction can proceed faster, and many more reactions can happen in the same amount of time. All right, so we call this the catalytic cycle, right? Because catalysis is using enzyme to lower activation energy. So a catalytic cycle is when you have a substrate and you have an enzyme, they bond together, they either form a bond or break a bond, depending on what kind of enzyme you're talking about. In this case, it's breaking a bond. And then the new products are released to go on their way, and you have a enzyme that is now available to be reused over and over again. So that's one complete catalytic cycle. So it's basically going through one whole reaction of using an enzyme, making the product, and then getting rid of the product. So factors affecting enzyme activity. We talked a little bit yesterday about how enzymes are a little bit like Goldilocks. They like this, they have a preferred optimal temperature and a preferred optimal pH. And it can be different for lots of different enzymes. But basically, for temperature, for this enzyme in particular, you have 40 degrees Celsius is its optimal. And if you keep things at 40 degrees Celsius, that enzyme can be working fine all day long. But if you get it up too hot, get it up here to about 65 degrees, that enzyme is just going to completely stop working. Same thing as if you get cold. You could take it down to 20 degrees. Now that enzyme is only working at about half capacity. So it really has a preferred range, and varying on either side of it is going to make the shape of the enzyme change, and that's going to make the enzyme not function anymore. So you can turn off the enzyme first by either changing the pH or the temperature so that they're no longer optimal. And if that happens, then you can shut down an enzyme. The cell can actually stop production of an enzyme and only make it when it needs it, so that that's one way it kind of can control it. And it can also use what we call inhibitors. Okay, inhibitors are a couple different kinds. You first have a competitive inhibitor and you have a non-competitive inhibitor. Okay, and we talked about these yesterday. We just didn't use these words for them. Okay, and I'm going to remind you here in a second. But basically, a competitive inhibitor means that something else. So you have your enzyme. You have this other thing that is able to bind to it impartially, basically blocking this substrate from being able to bond. So remember yesterday how when I was talking about the toothpick A's, I put that round ball in my hand? That was mimicking competitive inhibition, because if there is something in the enzyme, if I had something in my hand, then I couldn't perform the function my hand was supposed to be performing, which was breaking the toothpick. So the same thing's happening here. Something else is in this hand, and the substrate cannot bind, and so the substrate is not going to be turned into whatever it is it's supposed to be turned into. Okay? Non-competitive inhibition is basically something changing the shape of the enzyme so that the enzyme, even if it finds the substrate, it's changed shape in some way so that the substrate cannot bind. Um, lots of time this happens. We have what's called an allosteric site. Allosteric means shape. There's a site where something else can bind. So in this case, there's some sort of inhibitor here that binds to it. And because this binds, it changed the shape here. So now now this can't bind here anymore. So this could be result of some sort of feedback from the somewhere in the cell that says, okay, we don't need any more of this product, and it sends this molecule here to bind to the enzymes to turn them off. If this is removed, then the shape would return to normal, and the substrate could then proceed to bind. All right, so you can also turn on an enzyme, and there's a couple ways that this happens. You have what's called activators. So these activators, one way is that they bind to the allosteric sites and keep enzymes in an active configuration. So if you go back a second, here's the allosteric site, okay? This would have been an allosteric inhibitor, but you can also have an allosteric activator. And if the activator binds there, basically what that means is that it'll bind there and it'll keep the enzyme open and available and prevent an inhibitor from binding there. So it's a way to make sure that the enzyme stays open and functional. And then you also have things called cofactors or coenzymes. Cofactors are inorganic molecules, very small. Coenzymes are organic molecules, and they work with the enzyme most of the time in order for something to work. So this example here is a nice example because it shows you how without this cofactor or coenzyme, the substrate really wouldn't have fit in that enzyme. But because the cofactor fits in here first and fills in that gap, now this active site fits the substrate better and allows the substrate to fit. Okay, so this is a case where in order for that substrate enzyme complex to work, you need a cofactor or a coenzyme in order to kind of help that process proceed. All right, and finally, enzyme cofactors, they are sometimes they're assisted by an additional chemical compound. Coenzyme, non-protein, organic molecule serves as a cofactor. So basically, kind of what we already just said in this section here. Small organic molecules, they work with the enzyme in order to allow the process to proceed. All right, guys, so that's it for today, and we'll get started on photosynthesis and cell respiration in the coming days.